Hello, everybody. Welcome to our webinar. My name is Hornburg. I'm a Senior Director in Research and Discovery here at Seer. And today we will have two talks for our webinar. I will get started by talking about how we can achieve enhanced proteome coverage and analysis scale with our proprietary nanotechnology-based platform. And after me, Bruce Wilcox, who's VP for Proteomics at Proteomics, will talk about large-scale deep and untargeted plasma proteomics for multi-cancer biomarker discovery. Now, I will have 4 topics today. So for everyone who first time sees our technology, I will give you a brief introduction on how the Proteograph works, how the nanoparticle work in achieving unprecedented depths in proteome samples. I will also talk about and give you some examples of recently published studies, as well as studies that are currently under revision for publication. And the last 2 topics, I'm really excited about talking about some of the future directions that we can take the technologies. One of which is using machine learning and modeling to understand the complex nano-bio interaction. And the second part will be about tuning the Raman effect, and you will learn more about what the Raman effect is during our presentations here. But tuning that Raman effect to get even deeper into the plasma proteome, leveraging competition between proteins for composing a so-called protein corona. So for this audience, I don't think that I have to go much into details on why proteomics is exciting and very important to understand biology. But just as a brief reminder, if we, for example, compare the genomics information scale to proteomics, we all know that proteomes are way more complex because they are not only having your individual proteins, but we are talking about certain isoforms of proteins, proteoforms that can be post-translationally modified. And of course, the interaction between proteins are also information carriers in biology. So we are looking at a tremendous complexity that will give us, if we understand how to get into that information space, give us a lot of insights into biology. Now, the challenge with proteome samples in general, and in particular for plasma, is that life happens at a log scale. So that means that a minority of proteins make up a majority of the mass. So for example, if you look into blood plasma, about 22 proteins make up 99% of the mass and thus signal for your downstream detector. And that comes with a big challenge because most mass spectrometers that are used in unbiased proteomics are having a limitation in terms of the dynamic range that they can see. And with some tricks, you may get it to 6 orders of magnitude, but that is not capturing the entire dynamic range of biology in complex biosamples like plasma. And many of the interesting proteins, like the cytokines down here, are really hard to detect. Now, there are some ways to get deep into the plasma proteome if you spend a lot of time on it. Those deep interrogations of the biosample include usually high abundance protein depletion approaches, where you need to have an antibody that basically depletes that particular protein that is very high abundant, and it disturbs your signal and makes it hard to detect the low abundant ones. And on top of that, you can fractionate your peptides in order to basically have a better dynamic range, a more accessible dynamic range. Now, this is enabling you to get very deep, but it's not scalable. It's taking weeks for a few samples, and it's not something you can apply to large-scale studies. At Seer, we are combining and condensing the complexity of these deep workflows to something that is manageable and easy transferable to different labs. And we are leveraging a proprietary technology that is centered around nanoparticles. And in the simplest design concept, nanoparticles have a superparamagnetic core and are surrounded by a glass surface, a glass shell. And this glass shell can be modified and can be engineered in a way that it has a very distinct, very specific physicochemical makeup. Now, as you introduce those nanoparticles into a complex biosample, what happens now is that you assemble proteins based on their physicochemical property space in so-called protein coronas. And this assembly is extremely reproducible, and it can be engineered to an extent where you really define that what subset of the proteome you're capturing. And this is not a few, but it's thousands of proteins. So how does it now actually work? So when we introduce a nanoparticle into a complex biosample, like, for example, blood plasma, it starts to assemble these protein coronas, which are driven by two factors: the affinity of the proteins to the surface, as well as their concentration, which is important because you want to keep track of the concentration information in your biosample. Now, this leads to an effective compression of the dynamic range, in that some low abundant proteins will have a higher affinity than some other high abundant proteins. And you assemble per particle coronas of thousands of proteins. Now you can assemble a panel of particles with different physicochemical properties to interrogate the entire information space of a complex biosample. We have integrated this whole workflow and this whole concept into the Proteograph, which is coming with all you need to get from your biosample to the insights. So we start out with the consumables that contain all the peptides and all the particles that you need for proteomics interrogation. And we have integrated that into a liquid automated handling processing device, which takes you from your biosample to your peptides in roughly 7 hours. We are mass spec agnostic, so our technology will scale and integrate with every existing mass spec technology in your lab, as well as with every new mass spec generation that is going to be released in the future. We also have a data analysis pipeline that takes care of data normalization and raw data processing in the cloud, so it scales well from 10 to hundreds, if not thousands of samples. And also it includes a QC analysis part, which makes sure that you can trust your results, that they are comparable from today to tomorrow, from today to in a year from now, as well as cross labs. Let's have a look at some real data. Here we are seeing some data that we currently have in a revision for PNAS, publication in PNAS, comparing our Proteograph to conventional deep proteomics workflow and neat plasma. We used here a common pool plasma that was subjected to all of these different workflows, and we used the same short 30-minute DIA runs and Diane to analyze the data. What you see here is that we get close to 3,000 proteins in a single pool plasma with Proteograph, 1,500 with a depletion and fractionation approach, and around 800 to 900 with a depletion approach alone. Now, when we ask a question, where do we gain proteins? We see that we dive deeper into the proteome as we're using the nanoparticles, since they make that information more accessible to the downstream detector. Importantly, we also have to look at the precision, because if you just have high throughput and you essentially get more information, but the precision is compromised a lot, then what you will end up is having the requirement of measuring more samples, so that's eating up your throughput again. Luckily, what we see here is that we get pretty high precision with CVs less than 20% on average with Proteograph. And you have to keep in mind that this is across 3,000 proteins. So looking into the much less abundant signal of the proteome. And this compares pretty well to depletion, where we only get 1/3 of the proteomics information, but essentially the more bright signal. And yet we are still in the same precision regime. One additional important aspect, of course, is the response, the linearity of a response, because you want to make sure that you can actually observe biological differences in your biosample. So we did this spike in recovery experiment that was published last year as part of our Nature Communication publication, where we compared the intensity measured derived from a mass spec readout from the Proteograph to Seer, to an ELISA readout for the same biosample before subjecting the plasma to the nanoparticles. And what you see here is a linear response, a linear regression between the two signals, indicating that even for small changes in the proteome composition, we capture that information using nanoparticles. So here's the publication in which we published these results, in addition to using now a Proteograph to dive deep into some biological studies. This was a 141 cancer subject study where we used a random forest classification strategy to distinguish between healthy controls and NSCLC subjects. And you see we got a pretty decent performance here. And on the right-hand side, you see the most important features that contributed to the classification. And I would say this is a desired result that you have in proteomics or in omics in general, where you see some known targets mapped by open target scores in red here, and some gray proteins, indicating here some not yet implicated proteins for the disease. So you have a nice mix of confirmation as well as potentially new and interesting to follow up targets. Now, for the last two sections, I like to dive a little bit deeper into how we can now combine machine learning and our understanding about the nano-bio interactions to further drive our technology. So one way to do that is to drive our understanding of the intricate network of biointeractions, nano-biointeractions at the surface of nanoparticles. And it's always good to start from a simple approach. So for example, if we take 37 different nanoparticles and we just ask the question, hey, what are the different functional groups that we can classify as the functionalization of the nanoparticle surface? And a subselection of that, for example, looks like that. We have some nanoparticles that have carboxylate groups, we have some hydroxyl group, we have aromatic functionalization, we have some polymeric particles. And now we can start asking the question, hey, how does it associate with certain protein corona compositions? And here on the right-hand side, you see a heat map in red and blue showing the differential abundance of proteins in these individual coronas, again, like coming from a plasma proteome that we subjected to the nanoparticles. And you see there are some subclasses. For example, here we have amine and coordinating groups clustering with some specific protein corona compositions. Interestingly, when we transfer that to the level of annotations, so now asking, are there any protein groups and families, functional classes of proteins differentially interrogated by specific nanoparticles? Also here we see a differential clustering. So in red, we see differentially enriched proteins. In blue, we see differentially in depleted protein families. And you see, for example, that we have some proteins that belong to the adaptive immunity, immunoglobulins. We have some innate immunity-related proteins. We have some cytoplasma and membrane-associated proteins. And then we have certain particles that seem to be better in interrogating the subclasses of proteins. And on the right-hand side, we map what we call the fingerprint of these subgroups of particles, essentially showing like whether there's an over or under representation of certain chemical groups on the surface of particles. So that tells us that there's a really engineerable connection between the proteins we see and the protein families we see and the physicochemical makeup of the particles. We can take that one step further and slightly turn it around by looking now at the individual proteins and ask how much of the observed variation across the set of these 37 nanoparticles is explained by a particular functional group. And what you see here is a variance decomposition analysis following one line indicates a particular protein, and it explains variation across all the different nanoparticles. And we can now pick out a couple of proteins, for example, here the PTX3, which seems to be explained mainly by charge. And if we look at the isoelectric point of the protein and its predicted charge under the assay condition, it makes a lot of sense that given that it has a very negative charge, that it is actually driven mainly by a charge characteristic of a nanoparticle. Another example here is KLKB1, which seems to be not explained by charge at all, which is consistent with its isoelectric point pretty close to neutral and thus almost basically no net charge. But it is explained a lot by sugar moieties. And we see it has a PAN domain, and PAN domains are known to interact with sugars. So we can transfer that now to almost in some type of network analysis where we have in green all the points indicating proteins identified across all these particles. And then we have the nanoparticle properties, and we ask, like, within this experiment, how much of the observed intensity is connected to a particular functional group. And we have some proteins, for example, that are driven by aromatic functionalizations and polymeric functionalizations, and some proteins that are exclusive to some of these functional groups. So that gives us a machine learning way to further optimize and engineer nanoparticles to, for example, be focusing on a particular subset of proteins and protein families in the engineering process. So the last part of my presentation I would like to spend on talking about how we can further tune the Raman effect, which is the core of how nanoparticles work, in order to get increased performance and increased throughput. So just as a reminder, the Raman effect essentially describes the displacement of high abundant proteins on the surface, and thus the ratio compression that I indicated initially. So when you introduce a particle into your biosample, initially proteins bind essentially based on their overall concentration only. So you have basically all the particles basically surrounded by high abundant proteins. Now, if you give time to reach equilibrium, the low abundant proteins with a high affinity have the chance to displace some of the high abundant proteins with a lower affinity. And that leads to a more diverse composition of the corona. But again, it's important to note that it's not only affinity, it's affinity and the concentration that determines the exact composition. But that tells us that if we drive that competition a little bit more, we might be able to further diversify the corona composition, thus diversify the insight we get into the proteome composition in your biosample. So the simple experiment that we conducted here is to essentially increase the composition, the competition between proteins, and thus the Raman effect by limiting the surface more and more, providing more proteins that have to harshly compete for binding on the surface. We read out everything with a standard LC-MS-MS method, here a timsTOF Bruker system, and data was analyzed with Diane. I'm only showing Diane data here, but we also did an analysis max quant. The absolute numbers, if you go for different search engines, differ, but the overall conclusion is the same. Driving the competition here from left to right, you see that we get more and more proteins. On the very left-hand side, I indicated here neat plasma, which essentially means there's zero competition in neat plasma, right? Because there's no surface to bind, so there's zero competition. So it's approximately like going to infinity on the left-hand side. But you see the IDs here in neat plasma are way below 500 protein groups. And as we drive the competition, thus the Raman effect for each of these particles, we significantly increase the ID performance. Now, it is important to, as we indicated earlier, to keep in mind that the precision needs to be high, because otherwise you're sacrificing statistical power and your IDs might not be really helpful. So in order to evaluate how much we actually maintain or even improve the precision in the assay, optimizing the Raman effect, we did here this accumulation curve where we looked and asked the question, how many proteins do we aggregate with a below a certain CV threshold here, for example, 25%. And all of these proteins are, of course, 1% peptide and protein FDR controlled. And what you see here that you're actually not compromising the precision. Quite in fact, you are aggregating more and more proteins with a better precision as you drive the Raman effect. And the reason for that is probably more complex, but in simple terms, one of the contributors probably is that you just get a much better signal by basically getting the ratio compression slightly higher. You get a better signal for your downstream detector, which helps with the CV and the precision of the analysis. Now, one of the things that we are also really excited about is to see how we scale with new mass specs that are on the market. And here, in particular, when we talk about the Raman effect, and as you have noted, if we increase the competition for binding, right, one way to do that is essentially limit the surface more and more, but that yields you a little less peptides at the end of the assay. So it's very interesting for us to evaluate now how does it work, like on a very sensitive mass spectrometer like the Bruker timsTOF SCP, where we can work with much less input material. In this comparison, for example, we investigated a 40 nanogram injection mass versus a conventional 200 nanogram injection mass on the timsTOF Pro. It's not an entire apples to apples comparison because it's a slightly different HPLC setup, but overall what you see on the left-hand side is that we are on par or better with the timsTOF Pro in terms of the number of IDs we achieve by using 1/5 of the injection mass. So you see here neat plasma again, and then here our performance across the different competition states. And we reach more than 3,000 proteins in the end using a 60-minute LC-MS-MS analysis time. So with this, I'd like to finish and thank you for your attention. I hope I was able to show you how we use nanoparticles to really dive deep into the plasma proteome and how we can further extend the utility of nanoproteomics and the nanobio interaction, both in terms of using machine learning to understand how they interact, as well as driving the Raman effect to further increase the performance for individual nanoparticles. With that, again, thank you for your attention. I hope that you enjoyed the talk. Please visit our website, join our posters, and we're also hiring. So if you're excited about multiomics and proteomics, feel free to contact us. We are expanding the teams. Thank you so much and talk to you soon. Thank you again for listening to our talk. Before we move on to our next speaker, we have a small poll now and also at the end of Bruce's talk. Would be great if you can spend a few minutes and giving us some feedback.